Thank you very much. I would like first to thank Gressens for the very kind invitation. I also took the liberty to slightly change the title because I wasn't sure I fully understood what he was meaning, so I tried to make it a bit simpler. First of all, I have to declare one bias, one conflict of interest. It will be only brain, but hopefully for those who are not interested in brains, they will probably get some concepts which might be applicable to other organs. And the other thing, I won't be able to say anything bad about animal models because I think they're very important. I will try to highlight some pitfalls, but I think basically we need animal models. The issue of time is very important when you compare, as you was mentioned before, especially small animals like rodents, humans. Obviously, it's very different. And since we are looking at development, we have a moving target because every day or every week we have a different target in terms of size, sort of growth, maturation, etc., and molecular and cellular processes. So that's the first difficulty compared to the adult world, for example. And one key issue which is related to that is are we targeting the right window during development for the problem we are trying to address in humans? And so I will come back to this important issue. And if we can target the right window, is this window in this specific animal, is it faithfully reproducing what we try to model from humans? And I will go through 4 different potential pitfalls which are very important to remember when we use animal models. And finally, when there is an insult, is the brain or the cellular process we are looking at in terms of time, is it the same timing that would occur in humans, which is very important, for example, for neuroprotection, because if timing is very different, we might have the wrong idea derived from animal models. So first, the key issue is how do we target the right developmental window when we try to model a disease? And I will take the example of the oligodendrocytes in the white matter damage in preterm babies. These slides were kindly given to me by Stephen Bach. And as you know, oligodendrocytes in the white matter is probably a key target, a key cellular component of white matter damage of the preterm. And especially, we know now from human studies that through development, these oligodendrocytes grow from a very immature form, which is the progenitor, to a mature form, which produce myelin in between. These 2 stages, which are called pre-oligodendrocytes, are the most abundant in preterm brains. And so if we want to really have a model which faithfully mimic a human preterm brain looking at white matter, we have to find in animal models the same type of development where we also have mostly pre-oligodendrocytes. And so just as an example, if you look at humans, the pre-oligodendrocytes are mostly abundant between 24 and 32 weeks of gestation, which is shown on the top. If you look at rodents, it's between probably 2 days after birth to 5 days after birth. So we have clearly to identify for specific cellular or molecular mechanism what is the time window corresponding in the given species you are looking at to use as a model. And if you move, for example, to fetal sheep, you can see it's a very different window. It's from 70 to 100 days of gestation, so much larger window in fetal sheep compared to rats or mice. But again, we have to define exactly what's the correspondence. And the linked element of this is that there is not a given window for a disease. It really depends what is the mechanism or the cellular process you're looking at. If you're looking at synaptogenesis, for example, in human preterms, it might be a very different window compared to white matter damage. So it's really, you have to identify your question and then look carefully what is important in human tissues and then go back to animal models and try to identify the correspondence. So there is, again, not a single period forever for a given model. You have to really test your window every time you change the question. Once you have identified your window, you have to determine if the model you are looking at is kind of mimicking what is ongoing in humans. It's not because you have the right window that it's the same processes which are going to occur. And you have to remember that the time window is very different. It's a few weeks in rodents, for example, to several months or years of maturation for the brain in humans. And lengthening allows for increasing complexity. I'm going to show you 2 examples for that. On the left side, you have a developing cortex of a mouse or rat. And you can see at the bottom, there's a ventricle below the black line. I'm not sure you can see the arrow. No, doesn't matter. So at the bottom, the black line is the limit of the ventricle, and the red cells are the progenitors of the ventricular zone. And then on the top, above that, there are green cells, which is subventricular zones. And in rodents, you have 2 zones where neurons are produced during gestation. And these two layers of progenitors produce most of the neurons of the cortex. In contrast, if you look in humans on the right, you can see that again you find the red cells and the green cells. So you have the two same layers, but on top of them you have some orange cells, which are even more important in numbers. And this third layer is very important because this is where the expansion in surface is occurring. This is because we have a third layer of precursors or progenitors in the human brain. The surface of the cortex is much bigger compared to the rest of the brain. So through evolution, to allow an increase of size, and because also the gestation is increasing in time, this allows to have a third layer of progenitors, which really makes things very different. Of course, if you look again back to the mouse, you can see there are also a few orange cells, but they're very few compared to humans. So in a way, you can still use the mouse model to try to mimic the human proliferation of neurons, but you have to take into consideration that complexity is quite different. So it's still a good model, but with some strong limitation. Another important thing which is very different in terms of complexity and size is the subplate. And subplate neurons are very important, especially in preterm babies, because it's at the time of preterm delivery, the subplate is very huge in humans. You can see at the top, I will try to show you. You can see here, this is the white matter, and the whole thickness here is the subplate, is the cortex here. The subplate is 3/4 of the whole thickness of the cerebral wall in a preterm baby. So it's a huge structure where most of connectivity from thalamus is transiently connected over there. So it's very important for brain organization. And we know now that preterm baby have significant lesions in this subplate layer. If you look at rodents, this subplate is very tiny. This is a cortex of a rodent here, and you can see a subplate is only 1/5 of the thickness. So it's a completely inverted ratio. Rabbit is a bit better. Here's a cortex, and this is subplate, so it's about 50-50. But when you go through evolution, you can see the subplate becomes bigger and bigger, and of course, if you use a rodent, the role of the subplate might be underestimated and, in terms of outcome. One important thing as well is the impact of birth. Birth is in different species occur at different time point according to brain maturation, organ maturation. And birth is very important. In humans, you can see this is the rate of brain growth. You can see the round birth is a peak of brain growth. It's really a key event for humans, and this is where the brain growth is maximum. And if you look at different species, this is identifies the birth. Here's a black bar. You can see that the peak of brain growth can be very distant from birth, either before birth, like in monkey, or way after birth, like in rabbit. Only the piglet, which is here, kind of mimic the coincidence between birth and the brain growth. So whenever you consider different species, you have to remember that births occurring at different times, and especially in preterm birth, this might have a very important impact in the way you model your disease. And for example, if you look at synaptogenesis and different species, you have the right on the top, the cat, and monkey, and the man, you can see the profile overall is the same. So it's increased progressively and reach a plateau at puberty and decline with aging. But you can see that the birth time is kind of shifted to the right or to the left compared to humans. And so we think that birth is very important for impacting the developing brain. So depending on the species you are using, you might have a different impact of birth on synaptogenesis, for example. And just to show you a very clear example that birth might be very important, as you probably know, GABA receptors in the adult are inhibitory, but during development they are first excitatory because the chloride levels are different in the developing brain. So they shift from an excitatory status to progressively an inhibitory status. And at least in rodents, it is confirmed on human tissues. During birth, as you know, there is a peak of oxytocin coming from the mother, and oxytocin crosses the placenta and goes to the brain of the baby being delivered. And this oxytocin peak transiently inverts completely the efficacy of the GABA receptor. It becomes from excitatory completely inhibitory for at least a few hours. And so, of course, if you change the time of birth, this peak of oxytocin will be moved, and so you have a major impact on the efficacy of the GABA receptors, and this might have an important impact on the brain development. So, and this was even more important than just oxytocin. Hugo Lagercrantz a few years ago made a review looking at different neurotransmitters and this is on top humans, on the rodents, on the bottom humans. You can see that the peak of these different neurotransmitters is highly influenced by birth. So again, if you change the time of birth, you will have an impact of a lot of neurotransmitters, and we know these neurotransmitters are very important for brain maturation. Conversely, shortening of the size and the time of brain development can induce what we could call legitimate relationships. I'm just going to show two examples. On the right panel, you have human brain. You can see the cortex here, and this is hippocampus here. So you can see the distance between hippocampus and the sagittal cortex is very, very, very big. It's a few centimeters. So if something going on here, there is little chance that molecules will diffuse up to the hippocampus. In contrast, if you take a rodent, you can see the hippocampus is just below the cortex. So if you make a lesion of the cortex, for example, you might expect to have an impact on the hippocampus, which might not be relevant at all to the human situation. So you might induce relationships which never exist in the human because of the size is very different. Also timing, this is a schematic representation of the major events occurring in developing human brain. You have production of astrocytes, production of neurons, migration of neurons, apoptosis, and plasticity, differential plasticity. You can see they overlap, but still kind of discretely different, and they go on for weeks or months. If you take a rodent, everything is compacted. It's only a few days or a couple of weeks, and so sometimes you have relationship which never occur again in humans. You have two different phenomena which shouldn't be occurring at the same time, but just because we've compacted everything, you might end up with two phenomena occurring. If you impact on one, you might have an impact on the other one, which should never occur in bigger species. Conversely, if you increase the length of gestation and maturation of the brain, you also increase the size, and you can move from a few microns to several centimeters. It's shown here, for example, from migration of interneurons. As we know, interneurons are coming from the lateral ganglion eminence, and they migrate tangentially to the cortex. It's the same phenomenon, the same mechanism in rodents here and in humans, but the size is very different. This is the real size of the mouse embryo at the same stage as the human embryo. You can see there is a huge difference. The distance that the neurons have to migrate is hugely different. It's even bigger 5 weeks later. So exactly the same mechanism, but distance and duration completely different. And so, of course, there's much more chance to interferences in the humans because the size and the time is much longer than in rodents. And the same, for example, for myelination. Myelination in rodents takes place in about 2 or 3 weeks. It's completed by 3 weeks after birth. In human, it takes weeks, months, or years to complete. Up to adolescence, you still have some myelination ongoing. So again, it's very difficult to model a phenomenon which takes place in years while it only takes place in a few weeks in rodents. So the time difference is huge, so you have to take this into consideration when you try to interpret your data in animal models. So last question, I think very important as well. We do an insult, which a lot of us are interested in, is the response of the cells, of molecular cellular response in terms of time. Is it relevant or very different from humans? I think this is where the rodents are really important again, because for a lot of events occurring after an insult, it's about the same time window, and we'll show you just one example. After hypoxic ischemic insult, for example, in term babies, there is an acute cell death, which is mostly necrosis, but then there is a secondary latent phase, and then secondary energy failure, which is associated with apoptosis, and different type of programmed cell death, but mostly apoptosis and autophagic death. And this has been studied for many, many years, and the summary of all these studies is showing that after the primary energy failure, within a few hours, we have a latent phase, and then by 24 hours, it depends, between 16 and 48 hours in rodents, you have a secondary cell death. It's very conserved, depending on the severity of the insult, but roughly it's about 16 to 48 hours, you have secondary cell death. If you look in piglets, much bigger animal, you have the same thing. This is a study by Nicky Robertson using MRS, looking at secondary energy failure by 15 hours, so about the same windows in rodents you start to see secondary energy failure. If you're staining for cell death, you find secondary cell death within the same window. And the same in humans. This is mostly MRS, but also some postmortem data showing that, again, between 24 or 48 hours, you find secondary cell death. So despite the fact we move from rodents to piglet to humans, completely different size, completely different timing of brain development, the secondary cell death occurs almost exactly the same time. It's the same from inflammation, at least all the acute response to damage seems to occur roughly within the same time window. So even rodents seems to be relevant, well, at least when you look at the early response to damage. So to summarize, I think we have to remember that we are looking at developing organs, so it's very important to try to keep in mind that it's different from one day to another one, so it's a moving target again. Obviously, there are limitations with animal models. I just show you 4 of them, there are probably others as well, but so we have to remember there are pitfalls, limitations. We have to be extremely careful when trying to interpret our data and translate it to humans. This especially much more complex human brain and other organs are much more complex compared to rodents or even piglets. Burrows, this is probably the key issue, the most difficult to take into consideration. Burrows is very different timing in humans versus all the species we can use, and this is probably a very deep effect on what we are looking at. Also, you have to remember that sometimes, because everything is smaller, shorter in rodents, sometimes you have things which happens together or next to each other which are not relevant at all for humans. And also lengthening and sizing up also makes things much more complicated and with different impacts in humans compared to smaller animals. However, despite this limitation, I think we still need to use animal models. It is, I think, my conviction. And we shouldn't throw the baby with the water or the bath. And I think probably the way to use the best animal models is to combine them. Very often people ask, what is the best model? I don't think there is such a question or such an answer. I think every model has a lot of limitations, a lot of pitfalls, but if you combine multiple animal models, going from rodents to large animals like piglets or rabbits, and then to monkeys, non-human primates, if you find the same thing over and over again, it's probably meaning that it might be also true in humans. So it's very combination, which is very important. And just show you this paper, which is just an example where we try to think about what's the best way to combine different animal models in the field of neuroprotection to try to achieve the best candidate drugs to move to humans. But again, I'm really convinced that animal models are still valid, still useful, if you use them in the right way, and the right way is probably to combine as many as you can. So again, the best model is probably not the right and relevant question. And I will stop here and thank you very much for your attention and ready for questions if you have any. Thank you.